pasamos directamente al siguiente ponente.
Vamos a pasar a la segunda ponencia. Buenos días a todos. Vamos a hacer un cambio de tema. Vamos a ir a anticuerpos neutralizantes y yo voy a cambiar también de idioma. So, good morning. We are switching to monoclonal antibodies, broadly neutralizing monoclonal antibodies. We saw yesterday that these antibodies can be useful in the treatment and the prevention of HIV, that even the glycosylation status of these antibodies may modulate inflammatory responses. And today we are going to learn about the use of these antibodies on cure strategies. And for that, it's a pleasure to have John Frater with us today. John he studied medicine in Cambridge. He got his PhD in London, in the Imperial College, and now he's professor of infectious diseases at Oxford University. His research work is HIV cure, and he's actively doing basic research and clinical trials on this goal. He has participated in broadly neutralizing antibody trials, such as RIO, but also in cure strategies using broadly neutralizing antibodies. And he's going to tell us about what is telling us the clinical trials on broadly monoclonal antibodies about the HIV cure. Fantastic. So, thank you. John, please.
Thank you very much. So it's fantastic to be here. Thank you to the organisers, to my good friend Javier, for this invitation to come to this beautiful city. And we've had some great collaborations in the past between the UK and Spain, so it's nice to sort of keep that all going. I remember Maria's second slide that she put up there showed the list of European countries and some statistics, and I was trying to find UK, and then I remembered we're not there anymore, and it was kind of depressing. And a bit of a reminder of kind of some of the things we need to do better. But look, it's fantastic to be here. I'm going to be talking to you a bit of clinical data. I know there are many clinicians in the audience. I know there are also scientists. It's a bit of a mixture. But this was the translation of the title I was given, which I really like. What do monoclonal antibodies whisper to us about HIV remission? And I like this word, because I haven't seen this before. No, it's kind of, what do we learn? These trials are whispering to us. They're telling us their secrets. And there's something about a whisper, particularly in these sorts of trials. And what is a whisper? Well, a rumor or a piece of gossip, okay? So yeah, we're scientists. It's true, it's not true. But maybe we're not sure sometimes. And these trials, I think, are a really good example of sometimes we don't really know how to interpret the data. And BNAbs are a great example, because, we have antiretroviral therapy. We've just seen the data. It's fantastic. 100% protection. It works in people who take their therapy and are able to take it really well. The bar is really high. But people do want to be able to come off their tablets. You have the stigma of seeing your pod of tablets in the kitchen every morning and reminding you that you're living with HIV. You have the memory that if you stop taking your tablets, things might come back. We're doing really well, but we're not there yet. So there is a rationale to come off ARVs at some point, and to get a cure or long-term remission is still a real goal, I would argue. There's a lot of debate. Do we need to do this? Should we be investing money in this? But actually, you talk to the community, and this is something they still do ask for. So BNAbs, broadly neutralizing antibodies. Two really interesting questions. Are they good just as antiretrovirals, as drugs? We think they probably are. And that's fine. That's kind of, we can do that in clinical trials. We're quite good at that sort of thing. But there's this other question, which I think I want to talk to you a bit about today. Is there this, does something happen afterwards? After you have a broadly neutralizing antibody, do we see some protection once the BNAb has stopped working? And this concept of the vaccinal effect, which is pretty controversial. So this is an example. Many of you will have seen these sort of slides before. These are antibodies sticking to envelope, and you can see there's lots of different antibodies that have been thought about. These two groups in particular are where we're interested: the V3 loop and the CD4 binding site, and in fact 10.10.7.4 and 3BNC are the two that have been focused on. So I was asked to kind of go through a bit of the trials, and I was trying to think of a good way to do this. So I'm going to give a bit of a history lesson. And for some reason, I decided to use Star Wars as an analogy. I don't know why. It just seemed kind of appropriate. So we're going to start at the beginning. For the Star Wars fans, all the episodes are out of order, so don't get angry with me. But we're going to start with episode 1. And this actually just reminds us that over 20 years ago, we were already trying to do these trials. We were already giving antibodies to people. And this is Alexander Tricola, Huldrych Guntard in Zurich. They had 3 antibodies. They stopped antiretroviral therapy. We think we're doing this new now, but we've been doing this for a while. It kind of worked a bit. The antibodies aren't as good as the antibodies we have now. It's probably 2G12 that was the main player in this. And there was a little bit of a hint of control, but lots of immune escape. They weren't very potent. But it was kind of proof of principle that these antibodies might be useful moving forward. So that was the beginning. We thought, they're not good enough, but maybe in the future. And then this paper came out, and this is a methods paper, and it was basically saying we have a new method, a new way of discovering what the antibodies are that are circulating. And this was a baiting technique that Michel Nussenzweig's lab in Rockefeller in New York developed. And because of that, we could actually start to see which antibodies that were circulating could be protective. And it's changed our opinions of what was out there. These antibodies are really unusual. They've got very different structures. They've got big CDR3 loops. They're very mutated. They're biased to certain sort of arms, like a kappa chain and things like that. And that changed the way we thought about things. So there was a lot of optimism then. We thought, actually, maybe we can build neutralizing antibodies that are going to be effective. We can learn about it. But as with optimism, after that often comes disappointment, and the empire strikes back. And here, so there were clinical trials in VRC01. Many of you may remember this paper. This was Katie Barr's paper in New England Journal. So VRC01 was the great hope of an antibody that might be effective, but it kind of didn't really work. People stopped therapy, but the virus came back in 4 or 5 weeks. Now, that's not good enough. If we are going to make a big difference, we need to see substantial delays, because what we have already is so good. So that was a little bit depressing. But then not long after that, we saw this paper, which came out. And this was the Johann Steig's paper. And this is the first time that there was real evidence, I would argue, that we got a broadly neutralizing antibody making a difference. So this is 3BNC. It binds to the CD4 binding site. And here, in this example, we now see a much more significant delay in time to rebound when people stop taking their antiretroviral therapy, 19 weeks of delay. And that's starting to move into territory where we think this might be useful. So again, it's kind of proof of principle. There was escape, and that's something we need to worry about here. These variants do escape from these antibodies very quickly. But this was important. So this set the scene. And I apologize, the Star Wars is finished now. We can move on. So what came next? And then there was a flurry of studies. Loads of trials came out quite shortly after each other. First of all, there was the Mendoza study that some of you will remember. And this is putting 2 antibodies together now. So we kind of should have learnt from this, from when we were giving antiretroviral therapy back in the 90s. If you give 1, it doesn't work very well. If you give 2, maybe it works. You kind of need to give more. And obviously, 1 monoclonal wasn't going to do very well. 2 monoclonals made a big difference. And so this is Pilar's study, which shows that if the reservoir is sensitive, you get significant delay. Then there was a study from Michael Sneller working with Tae-Wook Chun. And again, 2 neutralizing antibodies in a treatment interruption study, significant delay when you stop taking antiretroviral therapy before your virus comes back. And then Christian Gabler published this paper not that long ago. Again, 2 broadly neutralizing antibodies. And what these data on the right show, rather than just looking at when does your virus come back, these data are looking how big is your intact reservoir, the meaningful reservoir. And this was the first paper that showed that the reservoir was lower in people who'd had broadly neutralizing antibodies than people who hadn't. And that is exciting, potentially, because if we have a therapy that now actually tackles the reservoir specifically, then that is moving us into territory where we can start talking about cures and remission and things like that. So that is interesting. So there's control of viremia off art, that has been shown, and there's early evidence of impact on the reservoir. And as a result of that, there have been even more studies. And I'm just going to show you a list now, but just to give you the flavor that there is lots going on. So in 2022, many of you will remember eClear came out, Roadmap, Beat2. So people are now starting to combine broadly neutralizing antibodies with other things. So pegylated interferon, romidepsin. Can we put a concoction of different things together to try and target the reservoir, get that lower, and actually lead to long-term remission? Again, these studies gave a signal, but they weren't in themselves enough. Titan came out, adding in a TLR9 agonist. That didn't do anything much in addition. So we're starting to learn what doesn't work, as well as what does work, which is always really helpful. Remember, this is tricky. This is difficult to do. So many things we try will not work. And then last year at CROI, 2 studies were presented. The FRESH study from Tumbiandungu from South Africa, which showed 2 BNAbs with a TLR7 agonist in the FRESH cohort, this amazing cohort of young African women. And then we also presented the Rio study, which I'm going to tell you about in a few minutes. Now, while all of this was going on, giving these antibodies to humans, there's lots of fascinating work going on in the rhesus macaque model, where obviously you can do a little bit more. And this, I just want to show you one study here, because this, I think, if you haven't seen this, this changed a lot of the way that we thought about things. So this was basically one of those clinical trials I've just shown you, 2 broadly neutralizing antibodies, but given to rhesus macaques who were on antiretroviral therapy, where the virus is suppressed, and then they did a treatment interruption study, exactly as you would do in a clinical trial. And this was intriguing because the data initially are a bit confusing. So what you can see here, so if I'm going to walk over here because it's easier. So this is where the antibody was given, and there's this tiny little line here where the antibody is working, where there's proper viral suppression. But then the virus comes back, and it comes back quite high. It looks almost like primary infection. Now, these animals didn't get any more treatment. They didn't get any more intervention at this point. And not all the macaques did this. But in some of these macaques, and this is 2 examples, the virus then starts to come down on its own, spontaneously, to undetectable levels. And it takes a long time. You know, it's nearly a year. This is days. Okay, which is a bit of a concern when you're designing human clinical trials, which we'll talk about. But it takes a long time for that to come down. But it stays down. And the question was, well, the antibody can't be working anymore now. Something else is happening. Why are these macaques controlling? And one of the questions was, well, is this T cells? And with a macaque, you can knock out a T cell. So you can give an anti-CD8 antibody and see what happens. And that's exactly what the investigators did. So here we have examples of where 2 anti-CD8s were given. First of all, they gave an anti-alpha. And alphas knock out your CD8s, but they kind of also knock out your NK cells, and they knock out your gamma T cells as well. And then they gave a beta, which is specifically knocking out your CD8s. But what's really interesting in this example is when you knock out the T cells, the virus comes back, and when that antibody wears off again, you get virus control. And you do it a second time, the virus comes back, and then you get virus control. And this, I think, was the first evidence that we were aware of that actually, if you give an antibody, you can then induce a T cell response that should be protective. And we've always thought T cells are important in HIV. If you look at elite controllers, it seems to be T cell driven. All the GWAS studies say it's coming from HLA class 1. This is T cells. But our history of actually doing something useful with T cells, of really protecting with T cell vaccines, has been really difficult. In fact, I mean, you guys in Spain are some of the few people who've really made a difference with some of the vaccines in your BCN studies, and I can see Beer Motta in the crowd there who's led a lot of that. So this is exciting. So maybe we can harness our T cells to do something useful. Breaking news. We didn't think of this. So someone called Theobald in Harvard in 1907 had already worked this out. Okay, so Theobald was going to try and protect guinea pigs from diphtheria. Okay, and he worked out that if you give them an antitoxin, it kind of works. But if you combine the antitoxin and the toxin, and you give both together, you get a really strong, sustained immune response. Now, Theobald didn't know what was doing this, but he worked out that there was some other induced long-term mechanism. And this is undoubtedly based on this concept that we're starting to learn about, that you're making it immune complexes, which you may have discussed yesterday. You're making an immune complex between the antigen and the antibody. That has been taken to either a dendritic cell or some other cell where you're getting Fc receptor binding, and that is then driving these new T cell responses. Can we harness this? Well, it's kind of really interesting. So taking this further forward then, taking Theobald's discoveries into what we know. So what evidence is there that there's a BNAB vaccinal effect? Because, if we are going to get into this territory where there's going to be a cure one day, and it's not going to be tomorrow or in 5 years, I mean, we are talking long term, because we are in these such early stages, and we've been in these early stages for a long time, and at some point we need to move forward. But I think we're getting evidence that we are heading in the right sort of direction. So what evidence do we have? Well, I showed you the macaque data, and I think that's pretty convincing from my point of view, that you're getting some T-cell responses that are inherently controlling virus. There is some clinical data. So this is data from Olsoga's group on the back of the eClear study. And they gave a single broadly neutralizing antibody, I think it was 3BNC. And what they see in this study is a slightly messy figure in the middle there, but what you're looking at is GAG-specific immune responses. And the ones that are in the dark hash, these are the ones that are in people who've had a broadly neutralizing antibody. So they've only had an antibody, they're undetectable, but their T cells are responding. The T cells seem to like having an antibody. And again, that's not enough on its own. It just shows that the body's doing something. But then nice work from Daniel Kaufmann in Switzerland, again, the Nissel paper in Nature Medicine, shows the same sort of thing. About 6 weeks after getting a broadly neutralizing antibody, you start to see a bit of a T cell response. And that's kind of when you'd see it in a vaccine as well. So what we're trying to do is we're trying to get the same responses from an antibody that we would get if we were to vaccinate somebody. And if we can see those same T-cell signals, then that could be quite exciting. And we can go one step further. It sounds like you talked a little bit about this yesterday. But actually, this FC engineering, we always worry about the top end of the antibody where it's binding, but the FC seems to be really important. And these data are incredible. And if we can translate that into something, that would be really interesting. So this is a mouse study, mouse being infected with a lethal influenza. And they were given a flu antibody to protect them. Okay, but they engineered the FC receptor with 2 different mutations that were thought to enhance T-cell responses afterwards. And they said, well, can we protect the mice? So this is trying to make nature even better than it's given us already, which you might be slightly worried about doing. But this is what happens in the study. So it's a bit complicated. So the grey line at the bottom, that's just if you give the flu to the mouse, and they die within 7 days, most of them have died. The isotype in black there, that's the standard anti-flu antibody. And you can see you're getting some degree of control. It's better than nothing. But these Gali, for example, the Gali is one of these FC mutant antibodies which enhances T-cell responses. You see 100% protection of the mice. None of them died in this experiment. If you knock out their T-cells, though, they go back down to wild type. So this is a really strong example that you can engineer these antibodies to give you even stronger T-cell responses. And this is now kind of being developed to see if we can take it into humans. So this is really exciting. And we've shown similar results in other infections as well. Right, this takes me on to the last half, or the last third of the talk, where I'm going to talk to you a little bit about the study we've been doing in the UK and in Denmark and with colleagues elsewhere on the Rio trial. And I wanted to finish off with this, partly because I probably know more about this than the other stuff, but also because I think it starts to give us a feel of where we might go with some of these long-acting antibodies. So this was a randomized placebo-controlled trial. We gave 2 long-acting BNABs, the same ones you'll be familiar with, in people with early treated HIV to see kind of how they would get on if we stopped their therapy. So just to remind you, the LS BNABs. So again, you may be familiar with these, but we know at the top end, at the variable end, they bind really nicely, either to the CD4 binding site area or the V3 loop. But if you mutate the FC region with these 2 amino acids, they recirculate through the neonatal FC receptor, which means that the levels of antibody stay really high. I've written a little note to myself about TGN 1412, because we were talking about this at breakfast this morning. You've got to be really careful with messing with the FC receptors. So if some of you may remember, the TGN 1412 was the, I think it was an anti-CD828 monoclonal that they gave to 6 young men in a clinical trial in the UK. They all ended up on intensive care unit within half an hour with a massive cytokine storm, all driven by FC-driven responses. So you start messing about with the molecular mutations at this tail end, and you can cause real harm. So we have to be really, really careful. And there are ways of people trying to sort of knock down some of these enhanced responses. People are trying to enhance these responses. But again, before we start putting these into people, we have to be really, really careful that we don't make any terrible mistakes. These mutations appear to be safe. And they have a dramatic effect on the half-life of the antibody. So these are data from the Rio trial. So on the left, we've got 10-10-7-4. On the right, we've got 3BNC. You can see that the dose, the PK after the last dose on the x-axis there in days. The green line is this magic number of 10 micrograms, 10 micrograms per mil, which is thought to be probably the therapeutic cutoff. It may be higher, but if we just stick with 10, and you can see that after a single dose, we're getting up to 300 days before we start hitting that 10. So these antibodies are at therapeutic levels for a long time after a single dose. So if the levels map into protection, then this is a really nice sort of way forward, and this works well. So, I mean, there's the question of can we use these for treatment, and then also kind of is there going to be some sort of remission cure type question afterwards. So, a bit about Rio. So these were all people with HIV, ideally with primary HIV infection, and then there was a couple who we brought in just to help finish off with high CD4 counts. But the idea was all with high early treated primary HIV, on therapy for at least a year, and good evidence of immune reconstitution. We wanted to make sure that CD4s were greater than 500, good CD4-CD8 ratios. We ruled out, because this was kind of a first study, we wanted to make sure it was as safe as possible. We ruled out anyone with significant co-infections or comorbidities. And this is a controversial area that some of you know about. There's a lot of pre-existing resistance to these antibodies, just in people who've never seen them. Maybe 1 in 3 people is already resistant. How do we test for that? There's no good test out there at the moment. There are different options available. We chose sequencing, but even by sequencing, only one of them you can actually do an interpretation for, so 10-10-7-4. You can't really sequence 3BNC at baseline and go, are you sensitive or resistant? So we ruled out people who we thought were definitely resistant, and we made sure PrEP was available for partners, because we're going to be stopping treatment, and actually looking after not just the participant, but the participant's partners is really important as well. And then we had viral rebound definition. So once your virus rebounded, what do you do? Okay, in the old days, we panic. We go, take your antiretroviral therapy straight away, your virus has come back. Okay, but if you remember the macaque study I showed you, it took 300 days for some of those macaques to control their own virus. So how do you design a clinical trial where you might need to leave someone in a viremic state for a year before the trial works? It's almost impossible. It's a terrible ethical conundrum. How long do you wait before you think you've answered your question? There's been a lot of debate and a lot of argument around this. I mean, there's a consensus that if you leave someone around 1,000 for 6 weeks, that may be okay. Some people do longer, some people do shorter. If you're really high, then you don't wait. You put people straight back on therapy again. And doing a treatment interruption study well is really tricky. This was our design, 2 arms. Arm A was the intervention arm. So people on therapy, they had their 2 antibodies up at the top there. Then they had a treatment interruption. We wait for the virus to rebound, and then they go back on therapy. Arm B was our placebo. We thought we should do this properly, do a randomized controlled study with a placebo arm, because most of the studies have been sort of single-arm open label. So they all had placebo, and then they had a treatment interruption. We thought that was a bit mean. All these people doing really well say, we'll just stop your therapy, you'll have some saline, and then we'll put you back on therapy and you can go home. So rather than just leave it at that, we had a second stage that with the placebo on, when they rebounded, they were given antibodies as well. Okay, but they went back on therapy for 6 months. And what we were trying to do here is to give 6 months of therapy while the antibody level drops, and then have another treatment interruption. And the idea here is if you show control at this point, it probably isn't the BNAb that's giving you your control. It might be T cells. And that was the argument. So we designed that to say, well, can we do an experiment in a way that will show you that actually there is some sort of vaccinal effect occurring? So what did we find? So we enrolled 68 people, randomized, median age 40, all men, which is a real disappointment. It was so hard to recruit women to these studies. This was not deliberate. It ended up all being men, just from the way it worked out from the sites we recruited at. Median of 5 years on ART, median baseline CD4 is really good. And we offered a second infusion to some of them after 20 weeks. So half of the people in the intervention arm got a second infusion. And what did we find? Well, primary outcome data we measured at 20 weeks. And at week 20, and I'll show you the figure in a second, but 8 people in arm A had rebounded, compared to 30 in the control arm. So there was a significant difference. 75% basically was still controlling 20 weeks after having been dosed. It took about 45 weeks to rebound compared to 4.6, so better than we'd seen before, which kind of fitted in with this LS antibodies lasting for longer. But interestingly, there's a little subgroup of people who 96 weeks later, 7 of them were still not back on antibodies, they're still not taking their tablets again, still doing well, and not taking any antiretroviral therapy, which I think is quite remarkable. So this is the data in graphical form. So this is the primary endpoint at 20 weeks, and you can see there's a significant divergence where the red line is the controls and the blue line is the people who are on the BNABs. Of course, if this was antiretroviral therapy, it would just be a big flat line along the top at 100% control, wouldn't it? So, I mean, we just have to remember, what we really are comparing against. And then this is 96 weeks. And you can see that there is a significant decline, but still there are some people controlling. Now, I just want to finish off by talking about the patterns of rebound and saying, do we see a vaccinal effect? I've only got 3 minutes, I'm conscious of that. So some people rebound really quickly. So these people are probably resistant already to the antibodies. Some people take a little bit longer to rebound. So it's delayed. This is probably the antibodies themselves working. And then we have this group of 7 who control, they haven't gone back onto ART yet. Now, some of them were bobbling along just around 1,000. 3 of them were completely undetectable for this period and remain undetectable. So we think there are 3 different groups. But was there a vaccinal effect? If you ask Dali what a vaccinal effect looks like, this is what Dali thinks it is. That's a T cell being injected with a vaccine, which is quite intriguing. And there are 2 questions. Do we see new immune responses? And do these responses translate to viral control? So a little bit of immunology data now for the basic scientists. So ELISpots, first of all. These are people who are undetectable, aviremic, okay? And what we see out to 36 weeks is an increase in immune responses. So they look like, they're behaving like they've been vaccinated, okay, based on their ELISpots. They're making new HIV-specific responses. If you look at proliferative capacity, it's not quite so strong, but again, there is this picture over 36 or 12 weeks, depending on what you're looking at, that there is a change in immune responses. There's an AIM assay as well that we also do. There's a hint. That wasn't necessarily so impressive. But overall, you're getting this picture that the T-cell immune response is being activated by something in people who are completely aviremic. Yeah, if you normally measure someone on antiretroviral therapy, you don't see any change at all. So there is something new happening here. This is what it would look like if they've been vaccinated. Other genes make the same sort of response. POL and NEF, we see some sort of signals happening there. And then if you want to look at what sort of T cells are fluctuating around, we see CD8 T cells, effector memories, we see Th1s. Again, there's a hint that they're being activated. And then you get these things called T-bet EMs. These are transcription factors that are consistent with an activated T cell population. And again, we see signs here, more evidence that these are being activated by the presence of the BNABs in people who are aviremic. And we have this shift, and the bottom dark red lines here, a shift to a gag response of all your immune responses. So this is all really consistent with an immune response that is being activated in people who are controlling. If you do single-cell work and you look at what do these T cells actually look like, well, this is before and after, and you can see there's a contour plot on the right here, which is getting denser, and that density consists of 2 populations. On the top right, it's the CD8 cells. On the bottom right, it's the MATE cells. So again, the MATEs was surprising, but certainly the CD8 T cells are doing something new here, which is really interesting. Right, and these T-cell clones are HIV-specific. And in some patients, it's crazy. This is a graph of the number of T-cell clones you have at baseline that are HIV-specific compared to after your BNABs. And this is just an example of one patient. There are hundreds of new dominant clones coming out after BNABs have been given. And when you map where these clones are binding, they are predominantly HIV-specific, GAG-specific clones that are coming out. So we're making strong, strong T-cell responses. Interestingly, if you're interested in HLA class 1, we see no association whatsoever, okay, which is kind of disappointing. We thought we might, but it's not there. But the T cells are doing something. So does this, last question, I've got 2 seconds, maybe a minute if that's okay. Last question, does this actually map into something useful? So this is a forest plot. This is my line of irrelevance. If you're on this side, this assay means we're going to rebound quickly. If you're on this side, you're going to go slowly. And what is intriguing here is that the AIM assay was protective. But if you make a baseline ELISpot response against HIV, you do worse, okay? We were not expecting this. So the immune system is telling us 2 different things. If we just look at those people where there's no evidence of baseline resistance at all, again, the ELISpot response at baseline is harmful, okay, which we think is probably a marker of an active reservoir that's more willing to rebound, whereas things like your proliferative potential is a marker of whether you can actually control. And you can make a score out of this. You can make an immune score and go, at baseline, are you going to do well on a BNAB or not? And based on that score, you can actually divide your cohorts in half and say, I can tell the people who are going to do really well on BNABs based on their baseline immunology. Right, I'm going to wrap up here. So the T cells are doing something when you have an antibody. We're not entirely clear what's happening, how they're talking to each other, or what the role of the autologous antibodies in innate immunity is, but it is certainly exciting. Next steps, we're going to put the BNABs together with a T-cell vaccine, and this is a study called ABVAX, which we have just opened, and we're going to combine T-cell vaccine with BNABs and see if that makes any difference. And to finish off, what have the whispers told us? So BNAB infusions, they do induce new T-cell responses. The question is how and why, and are they useful? These T-cell responses do appear to contribute to control, which I think is really encouraging, in these early steps, in these foothills where we are now, to think, are we going to have a cure in 20 or 30 years' time? It does look like we can harness the T-cells. But what's weird is that it is the baseline response, before you even have your BNAB, that defines how you're going to do. It's not how your T-cells change afterwards. And if you're from pharma, the target product profile, this is what has been decided, TPP for a remission agent for HIV. Viral load under 200, 20% of individuals can achieve it. This is the minimum. 10-year remission and over 10-year relapse rate remission for 2 years. So we're closing in. We're not there yet, but we are now closing in on that, and I think that is encouraging. So Rio is a massive operation. Huge thanks to so many people who've taken part. And I should also say we've got ARM-C, which is opening out, which is being opened in Barcelona as well. So it's great to have a Spanish team on board as part of that. And this is my last slide, because these are the people actually, I just stand up at these podiums and do all the talking. These are the people who actually do all the work. And they're an amazing group of mine in Oxford, so I'm delighted that they do all of that. And also thank the participants. I mean, what these guys have done, the armsfuls of blood and biopsies they've given us is amazing. So I will stop there, a few minutes over, I apologize, but thank you. Okay.
Thank you very much, John, for sharing these whispers with us. Unfortunately, we have no time for questions, so if you have an urgent question to John, you can just talk to him at the corridors. So thank you very much, both speakers, for this excellent session, and I hope you will have an excellent meeting. Thank you.